Okay, well, I might as well get going with the introduction. So welcome everyone to the SGP's calcium isotope proxy primer. Many of you are familiar with SGP, but for those of you who aren't, we are an open community-oriented research consortium that's aiming to aggregate and then analyze and understand the environmental evolution across Earth history. And as part of that attempt to better analyze and understand the record, we've started this Proxy Primer series, which is an attempt to accessibly introduce different proxies that we use in understanding the geochemical record. If you'd like to check us out, please visit our informational website at sgp.stanford.edu or our search website at sgp-search.io, which has our new phase 2 data release up. This data release includes 129,000 samples with more than 4 million analytical results that's been built by greater than 200 collaborators worldwide. And if you're interested in joining the project, please get in touch with us. So one of the things that we've been focusing on phase 2 is better incorporating carbonate geochemical data. And as part of that, I'm delighted to introduce Dr. Anne-Sofie Ahm to give today's proxy primer, who is one of the people that have been really pushing forward and a world leader in developing calcium and magnesium isotopes in carbonate rocks. So Anne-Sofie did her undergrad, master's, and PhD at University of Copenhagen before moving on to a postdoc at Princeton, where she really dug into the calcium and magnesium isotopes before moving out to start an assistant professor position at the University of Victoria, which is where she is now. So thank you very much, Anne-Sofie.
Thank you for the introduction. I will attempt to share my screen. Okay, I hope you see my slides.
Yep.
Okay, so I know this talk was advertised as a calcium and magnesium isotope proxy primer, but I have rudely crossed out magnesium because I just don't think I have enough time to also talk about magnesium, and I feel like magnesium also deserves its own proxy primer. So hopefully that's something we can do at another point in time. So why calcium isotopes? So it's definitely my favorite isotope system, and I hope by the end of the talk, it will be your favorite isotope system too. There's tons of applications for calcium isotopes, and I will really only touch on a few of them today. So we can use calcium isotopes to investigate the chemistry of bulk carbonate rocks for both paleoclimate reconstruction, investigating the global calcium cycle, and carbonate diagenesis. But calcium isotopes are also used widely to better understand biomineralization and mineral precipitation. It's used to investigate other biomaterials, so plants and animals, specifically looking at trophic level effects. It's also used in high temperature geochemistry a lot. So if you Google calcium isotopes, you won't just get carbonate papers. You will actually get a lot of papers that look at continental and ocean crust and mantle evolution, etc. And it's also used in cosmochemistry. So people use it for nucleosynthesis, thinking about the evolution of our universe. And it's also used in biomedical applications. So actually, you can use calcium isotopes to detect bone loss in astronauts. And it's one of the earliest proxies you can use to detect that kind of breaking down of your bones when you return to Earth after having been in space. So tons of cool applications. Today, though, I will only really focus on the top one here. So I'll talk about bulk carbonate geochemistry and how we can use calcium isotopes to better understand this. So calcium is a major constituent in most carbonate minerals. And here I've just listed some of the more common ones. So you see here, calcium is a main constituent. So in calcite, for example, 40% of this mineral is calcium. Here I'm also showing the crystal structure of some of these different minerals. And I have also listed the calcium coordination number. So what this means is that in these different minerals, in these different carbonate minerals, calcium is sitting in the lattice in slightly different ways. So the coordination number is the closest neighbors to calcium. So in calcium carbonate, calcium is ionically bonded to carbonate. And in calcite, it has 6 oxygen isotope atoms. No, just oxygen atoms sitting close to calcium. Whereas in aragonite, there's actually 9. And I included this little link in QR code if you're interested, where you can actually open up these crystal structures and rotate them around and kind of see this the different coordination numbers. And I'll come back to that later in the talk because it turns out this might actually be important for how calcium isotopes fractionate into different carbonate minerals. So the calcium isotope notation, when we talk about calcium isotopes, normally we talk about the delta 44 value, which is the measure of the concentration of calcium 44 relative to calcium 40 in a sample relative to a standard, and then in promille notation, so -1 times 1,000. Here I also have a table where I've listed the abundance of the stable isotopes of calcium. And if you look at the top row here, you would see that calcium 40 is by far the most abundant isotope. So 97% of all calcium is calcium 40. And the second most abundant isotope is 44, so about 2% is calcium 44. So one thing that's really confusing about the calcium isotope literature is that we don't always report calcium 44 to 40 values. You will find that some studies actually report 44 to 42 ratios instead of 44 to 40 ratios. And if you're not sort of aware of that when you're reading the literature, you could be comparing very different numbers. So that is confusing. You can relate these two to each other by assuming mass-dependent fractionation and use a kinetic isotope fractionation law that will tell you that the calcium 44 to 40 ratio is around 2 times the calcium 44 to 42 ratio. The second thing that's really confusing if you look into the calcium isotope literature is that we also don't agree on the reference standard. So again, you can very easily be comparing apples to bananas because people just report these numbers differently. So in this talk, I'll be talking about the delta 44 value measured relative to modern seawater. So that's my standard here. So that means that modern seawater has a calcium isotope value of 0 per mil. But if you look in the literature, you'll find that some studies report calcium isotopes relative to bulk silicate earth. So bulk silicate earth is around 1 per mil, -1 per mil relative to modern seawater, or more precisely, -0.96 per mil. Another really common standard is this NIST SRM 915A, which has a value that's -1.88 relative to modern seawater. So if you look up a calcium isotope paper, I will encourage you, for the first thing to do before you even read it is to figure out what is the notation, so you don't confuse yourself unnecessarily. So how do we actually measure calcium isotopes? So I made a little flow chart here. The first thing you're going to do is collect your sample of interest. That could be a carbonate rock, which is mostly what I'm looking at, but that could also be seawater or any kind of water, plant material, whatever it is you're looking at. You have to, if you collected a rock, drill out powders. So you have to cut and polish this rock and drill out powders. And then you have to dissolve these powders in a weak acid. So we try and leach out the carbonate component using buffered acetic acid. Luckily for calcium isotopes in carbonates, because it's a major element, we don't have to worry too much about leaching other components, which can be a really big issue for other isotope systems. So for example, if you're looking at lithium isotopes, you have to be really careful that you're not accidentally leaching clays that contain a lot of lithium. For calcium isotopes in carbonates with 40% calcium, we still leach out the carbonate component, but we really don't have to worry too much about it in practice. So once we have our dissolved sample, we're going to purify it. We are lucky here at UVic, we have this automated column chromatography system or IC system where we can put our sample in and it will push it through a column and actually give us a live readout here of what is coming through our column. So we have this conductivity detector that will tell us when our calcium is eluting through the column, and then we can actually collect that calcium peak. And then once we have our calcium collected, there's a little bit of treatment, and then we bring it into the mass spectrometer lab and measure the calcium isotope values. So there's two common methods to measure calcium isotopes. One is using a multi-collector ICP-MS, and the other one is using a TIMS, so a thermal ionization mass spectrometer. And I use an ICP-MS, so that's the technique I'm most familiar with. Each of these approaches have some pros and cons. So for the ICP method, the carrier gas inside this mass spectrometer, and I'm showing you a sort of a sketch of this here to the right. So the carrier gas is argon, and argon has a mass of 40. And that's a problem when you want to measure calcium-40. So unless you have a collision cell and sort of a fancier mass spectrometer, we will have to instead measure calcium-44 and 42. So this comes back to this weird thing in the literature where some studies report 44 to 40 and other studies report 44 to 42. It actually comes down to the differences in the analytical methods. But as I mentioned before, we can convert that to 4440, assuming mass-dependent kinetic fractionation. One of the good things about using an ICP is we can do a really high sample throughput. So we could easily measure 30 samples a day. We can use sample standard bracketing methods. And the precision is around 0.15 per mil 2 sigma, which is a lower precision than what you will see using a TIMS. So if you use a thermal ionization mass spectrometer, so the difference here is just the way you ionize your sample. In an ICP, you have a plasma, 10,000 degree Kelvin, that ionizes your sample, but in a TIMS, you're using thermal ionization instead. And that means you have a much lower sample throughput, maybe 3 to 4 samples a day. You have to use a double spike technique, but you can get a really high precision. So I think on average, maybe 0.04 per mil 2 sigma, although some studies report even lower precision. So depending on your research question and what you're trying to figure out, you have to consider which of these methods is more appropriate. Do you need a lot of samples with maybe a slightly lower precision, or do you need a few samples with a really high precision? Okay, so let's talk about the global calcium cycle and start to think about what can cause changes to the calcium isotope of seawater over time, the calcium isotope value of seawater over time. So the main sources of calcium to the ocean is continental weathering and hydrothermal inputs. And the only real sink is the burial of carbonate minerals. There are other minor sinks, but this is the main sink by far. So we could write up our sort of traditional mass balance equation if we wanted to, for example, create a model that would show the change in the mass of calcium in the ocean over time. So that will be equal to the sources minus the sinks. So F here stands for flux. So I have my weathering flux, my hydrothermal input flux minus my carbonate burial flux. And a quick side note here, if you look at this cartoon, you will see that the calcium cycle is really closely linked to the magnesium and carbon cycle in the ocean through this weathering of carbonate and silicate rocks and the burial of calcium carbonate. I won't talk much about it today, but it is worth noting that. Let's start to think about the isotope mass balance. So calcium has a residence time in the ocean about 1 million years, and it's well mixed, so it's homogeneous. If you go measure the calcium isotope anywhere in the open ocean today, it's going to be basically the same, 0 per mil on my scale. So on long timescales, we can assume that the input of calcium to the ocean reflect bulk silicate Earth. And as I mentioned earlier, bulk silicate Earth has an isotope value that's around minus 0.96 per mil relative to seawater. So in other words, our input from both continental weathering and hydrothermal inputs is lower than seawater itself, around -1. And then our sink, our carbonate burial sink, is isotopically depleted relative to seawater. So calcium 40 preferentially goes into the carbonate mineral lattice relative to 44. So seawater is heavy relative to carbonates. So we can also derive an equation for the change in the calcium isotope value of seawater over time. And when I give a similar lecture in my upper undergrad class, I make them derive this equation. But I'm not going to make you do this today. If you're watching this on YouTube, you could consider pausing here and thinking through the math if you can derive this equation on your own. The way we do it is we go back to our mass balance equation for the mass of calcium in the ocean, and then we multiply each term by its delta 44 value. So we have here this first step of deriving this equation. The next step is we now want to isolate the delta 44 calcium seawater over time. To do that, we have to use the product rule of calculus. So you can go through these steps on your own, but you will end up with an equation that looks something like this. So you'll see on the right-hand side, we have our inputs multiplied by the difference between our isotope value of the inputs minus seawater. And then we have our sink term, which is the burial of carbonate minerals, times the difference between these carbonate minerals and seawater. And we can also call this the fractionation of these carbonate minerals. So that's why I'm using this capital delta notation here. So I'm going to take you through a thought experiment, and that is what would happen to the calcium isotope value of the ocean if we doubled the weathering input flux. For example, if we have an increase in weathering rates or if we have ocean acidification. So I'll give you 10 seconds to think through this. So we are going to be inputting more calcium, which has a value around -1, into an ocean with a value of 0. What do you think will happen to the calcium isotope values? We could use our equations that we just derived if we wanted to actually calculate this. And if we do that, we could create a little model to show this change. So in this case, I have 2 lines on this plot. The x-axis is time, and the red line is the calcium concentration in seawater. So you'll see that the calcium concentration increases because we're now putting in more calcium. And you'll see that the blue line here shows the calcium isotope values of seawater. And we have this negative excursion where we have a decrease followed by an increase. So this is showing this increase in calcium inputs, which has a low isotope value, dragging seawater lower. But our system will respond by precipitating out more carbonate minerals, which also has a low calcium isotope value relative to seawater, which means that seawater will recover back up to 0 per mil. So we can take this thought experiment one step further and ask ourselves what actually would happen to the carbonate saturation state in this scenario. So in the simplest possible scenario, all we're doing is just increasing our calcium input to the ocean. We're not doing anything else at all. And if we do that, we'll see that our saturation state of the ocean would increase like crazy. So this is omega, where omega 1 would be perfectly saturated, and anything above 1 would indicate that we have supersaturation of carbonate minerals, and we see it goes to above 60. So that doesn't seem right. And the simple model that I'm showing you here is probably not really capturing this carbonate compensation feedback accurately. So we have to rethink this model. And I will say, if you go back sort of 20 years into the calcium isotope literature, this is sort of where we were. We hadn't thought through this next step of how should we actually model this carbonate compensation feedback. So There's some model improvements we could apply. First, we could think about if we increase the input flux of calcium, how would that impact our carbon cycle? For example, if we have ocean acidification and dissolution of carbonate minerals, we would expect that we are not only increasing the calcium concentration, but actually we're also putting more DIC or carbonate ions into the ocean. The other thing we could do is we could actually directly relate our sink, our carbonate deposition, to the carbonate saturation state, so 2 omega. If we do that, we can repeat the same exercise and we'll see some differences. First of all, our omega value doesn't go to 60 anymore, so that's good. We also see that we see kind of a muted response in both the increase in the calcium concentration of seawater and in the isotope value. So in this case, I'm actually doubling the input flux, and I'm only seeing a 0.03 per mil change in the calcium isotope value of seawater. I've included a list here of some really useful papers that kind of goes through this exercise that at least I personally find very useful for understanding this. And sort of the main takeaway here is that the calcium concentration in the ocean is really strongly buffered by this carbonate compensation feedback. And our calcium isotope values of seawater are also really strongly buffered. So they're not sensitive to changes in global fluxes. As I mentioned before, this is a doubling of the input flux, and it's giving you a change in the calcium isotope value of seawater that is below my detection limit on my mass spectrometer. So the second thought experiment I want to take you through is thinking about what will happen if we decrease the calcium isotope value of our bulk carbonate sink. So for example, if we change it from being -1 per mil to being -1.5 per mil. So again, I'll give you 10, 15 seconds to think through this. In this case, I'm not changing the fluxes at all. We're just changing the value of the sink, and we're making it more depleted. Okay, so this is what would happen. So this is the same type of figure as before. And because I haven't changed my fluxes, you'll see that the calcium concentration in seawater in this scenario is constant. But we do see a really pronounced increase in the calcium isotope value of seawater. So essentially, because we have changed our sink by 0.5 per mil, seawater responds by becoming 0.5 per mil heavier. So that our bulk carbonate sink ends up having a value relative to seawater, having a value that is equal to bulk silicate earth, which is our input value of -1. So in must equal out. So we can see that seawater can actually change. It's not like the calcium isotope value of seawater cannot change, but it does not respond to changes in or imbalances in global fluxes, but it will respond to, for example, a change in the isotope value of the carbonate sink. So we can also look at this in a steady state. So in this case, there will be no change in the isotope value of calcium over time. So I've taken my equation we derived earlier and set it equal to 0, no change over time. We set the input equal to output. So these fluxes will cancel each other out on each side of this equation. And then we can derive an equation for the calcium isotope value in seawater. And we see it's equal to the input value minus the fractionation for carbonate minerals. And so this is what I'm plotting here on the right. It's this equation here. You have kept the input value constant equal to bulk silicate earth around -0.96. And if we look at modern seawater, it has a calcium isotope value of 0. So we see that that implies that the fractionation of carbonate minerals today is around -0.96. So has the calcium isotope value of seawater actually changed through time? So here I'm showing you a figure from a study from 2016 where they measured the calcium isotope value of fossil corals throughout the Cenozoic. So the red values, or the red circles you see here, are these coral measurements, and they're comparing it to calcium isotope values in other archives as well. So the y-axis here in black is the inferred calcium isotope value of seawater. So we have the modern here, and we see we have a value of 0 per mil. And as we go back in time through the Cenozoic, it seems like there's a decrease, and our calcium isotope value actually goes down to around minus 0.5 per mil. So this is not just in the coral archive we're seeing this, it's also in these other archives as well. So this observation could imply that the isotope value of the bulk carbonate sink has gradually decreased as we go towards the modern. Or in other words, as we go back in time, the carbonates must have been less fractionated or heavier. So I can plot this on my figure from earlier. So if we go and look at pre-Cenozoic seawater, we have this value of minus 0.5 per mil. And that would imply that our bulk carbonate sink has a value close to minus 0.5 per mil at this time. So what could cause this change? So the paper I showed you before speculate that maybe this is related to the evolution of the pelagic carbonate sink. So the evolution of carbonates that could be deposited in the deep ocean relative to shallow water carbonate platforms. And I'll come back to this concept later on in this talk. Another thing you might ask when you look at this record is, why is it so messy? It's not the nicest trend. There's a lot of scatter and some differences between these different records. So to answer that question, I'm going to take a step back and just show you some data from the modern of calcium isotope values. So this is a compilation here of calcium isotope values in modern and recent carbonates from a review paper from 2020. And the first thing I want to draw your attention to is the scale. So if you look at the x-axis, we're going from 0 per mil to minus 2.4 per mil-ish. So there's a really large range. So what could cause this range? If you had to come up with some guesses, you might guess maybe temperature is important, maybe biomineralization is important. For example, if you look at some of these values here in skeleton calcite, bivalves, brachiopod, mollusks, there's a really large range in the modern. Similar, if you look at pelagic carbonates, forams, coccoliths, and dinoflagellates, there's a really large range. Maybe mineralogy is important. Here you can see that I've colored these different samples by their strontium concentration, and aragonite has a lot higher concentrations of strontium, so it stands out here in yellow and green colors. And it seems to be on the lower end of the spectrum. And you may also ask, maybe precipitation rate is important. And finally, maybe diagenesis is important. So I will quickly step through these different ideas. Is temperature important? So if you go back 25 years, when we started looking at calcium isotopes, it was actually thought that maybe this could be a really important proxy for reconstructing paleo temperatures. We have later found out that maybe this is not actually that important. So the figure I'm showing you here is from a paper from 20 years ago or more, where they're comparing different studies, culturing different forams and looking at their calcium isotope value and their sensitivity to temperature. Most of these studies show a very weak temperature dependence, where we have around 0.015 per mil per degree Celsius. And a few studies stand out as having a higher temperature dependence. And it's later been discussed that maybe these studies actually were struggling with with disentangling the effects of temperature and precipitation rates. So to sum up, we don't think temperature really has an important effect on the calcium isotope value. It's minor. Biomineralization, as we saw before, can have a really large effect on the calcium isotope value. So yes, this is important. Vital effects and species-specific isotope fractionation is really well documented. This is just one example from one paper that shows this, so measuring calcium isotopes in a bunch of different species. And we see this range both within each species and certainly between different species. What could cause this? A bunch of different mechanisms have been suggested. Maybe we are thinking about relay fractionation in the calcifying fluid, biologically mediated ionic transport, variable calcification rates, etc. What about carbonate mineralogy? Yes, carbonate mineralogy also seems to affect the calcium isotope value. So this is a figure from a study that showed that maybe this difference in the calcium isotope values of different carbonate minerals could be related to the calcium coordination number in the carbonate lattice. So in general, what you see here is higher coordination number suggested to result in more fractionation. So aragonite, for example, is isotopically depleted relative to calcite. On average, aragonite is around minus 1.5 per mil depleted relative to the fluid, whereas calcite is maybe around minus 1.1 per mil. What about precipitation rate? Yes, that also influences your calcium isotope values, and it actually has a really strong effect. So there's been a bunch of studies showing this, and this is just a figure from one of the more recent studies. So on the x-axis here, you have the growth rate. So towards the right, we have high precipitation rates of carbonates, and on the left, we have low precipitation rates. So you see at higher rates, we have more fractionation, so more negative calcium isotope values. If you were to extrapolate these data and ask the question, what would be the equilibrium fractionation when we are not affected by rates, you can see that maybe it would be pretty close to 0 per mil. The same relationship is also observed if you look at the strontium concentrations. So this is, I apologize for the resolution of this figure, but this was an experiment where they both looked at the fractionation of calcium, which is showed here on the y-axis, and also the concentration of strontium. So higher precipitation rates not only result in low calcium isotope values, it also result in high strontium concentrations, and vice versa. So when we go back and look at this compilation of modern data where we see this huge range, temperature has a minimal effect, but biomineralization is definitely contributing to this range. Mineralogy is contributing, precipitation rates is really contributing. So actually, inorganic calcite that I've plotted here, this is from experiments in the lab where they varied the precipitation rate. And you will see there's a 2 per mil range here just by varying the rates. And so diagenesis is not really affecting any of the samples shown here in this modern compilation, but certainly it could affect ancient carbonate samples. And this is specifically due to this relationship between the calcium isotope fractionation and carbonate mineralogy and precipitation rates. So if I had to sum up how diagenesis could affect the calcium isotope value, I've made this sort of simplified cartoon where we have primary carbonates that form in the surface ocean. And this happens on a relatively short time scale. So we're talking like days or over a diurnal cycle where we're forming maybe our aragonite on the Great Bahamas Bank. And these are relatively fractionated, so averaged minus 1.5 per mil relative to seawater. We might also form some calcite, which is a little less fractionated. Then once we bury these carbonates on the seafloor and they start to recrystallize during diagenesis, this happens over really long time scales. So precipitation rates in the pore space are very slow. So what that means is our calcium isotope fractionation factor becomes much smaller and it approaches 0 per mil. So we'll talk more about how this can affect some of these records. So I'm taking you back to the Cenozoic record here, where this is actually really well controlled for diagenesis. So these coral samples were screened for diagenesis. They were controlled for the mineralogy, so they're all less than 1% calcite. And we could also control some of the biological effects by picking out the correct or the same species. But when we go back in time, so this is now a record going all the way back through the Phanerozoic, and it's from a paper from 2007, it becomes really hard to control these different factors. And I will say, I don't think anyone has attempted to make a calcium isotope record since this paper in 2007, because we're starting to realize that it's actually really hard. We have to control for all these effects. And just to remind you, the range we see in modern carbonates is basically the entire range we see in this record. So I want to show you a case study where I want to talk about how we can actually start to disentangle some of these effects, and specifically thinking about calcium isotopes and diagenesis. So this is the Great Bahamas Transect. It's a number of cores that are taking from the shallow waters in the Great Bahamas Banks and into the Straits of Florida. And this is just a cross section showing you where these different cores are. One thing that's interesting in these cores is there's a change in the carbonate mineralogy with depth. So here I've just zoomed in on 4 of these cores that are taken from the slopes of the Great Bahamas Bank and into the Straits of Florida. And this brick symbol here is showing aragonite. So the top 100 to 200 meters of these cores are dominated by aragonite that is shedded from the bank top and transported into these deeper waters. And as you go down into these cores, you see they're mostly gray. So this is low magnesium calcite. So probably what's happening here is this aragonite is metastable, and over time it is recrystallizing and turning into a more stable mineral, which is low magnesium calcite. One thing that's interesting about these cores is if you look at the percentage of aragonite, you will see a very strong correlation to the carbon isotope values. So this paper here talks more about this relationship, but we see a change in carbon isotopes from 5 to 0 per mil. Okay, so here I'm just showing you one of these cores, and I'm showing you not just the carbon isotope values, but now also the calcium and strontium concentrations. So calcium isotopes and strontium concentrations. And one thing you'll see is as aragonite disappears, as we go down these cores, not only is our carbon isotopes going to lower values, we see a loss in strontium. So as this aragonite is recrystallizing, it's losing strontium and turning into calcite. And we see an increase in our calcium isotope values. So they go from this aragonite value of minus 1.5 towards heavier values in the first sort of 200 meters. So this is not just because this aragonite is turning into calcite. So here I've drawn on what would we expect if we just have a change in mineralogy. We actually see that our calcium isotopes is becoming even heavier than what we would expect for calcite. So what could be happening here? So think more about that. I'm going to show you the pore fluid profile now from the same core. So A here shows you the pore fluids in colored symbols. So we start at a value that's equal to seawater at the top of the core. And as we go down through the core, we see that the calcium isotope values of these pore fluids become lighter. And the white symbols here is the calcium isotope values of the sediment. So down the core, these pore fluid values become lighter because we're dissolving aragonite. So aragonite is dissolving, and the pore fluids is absorbing these low calcium isotope values. This aragonite is reprecipitating out as low magnesium calcite. And because this takes place over thousands to millions of years under really slow rates, there's essentially no fractionation when this is happening. So our calcium isotope values of this newly formed diagenetic calcite basically is the same as the seawater or the pore fluid in the core as we go down. So we can talk about different diagenetic regimes as we go down this core, where we have our primary aragonite in the surface of the core. As we go down, we see we have a fluid buffered region where our newly formed calcite is approaching the value of this pore fluid or this incoming seawater. But we also have a sediment buffered region where these carbonates have also recrystallized from aragonite into calcite, but they're not getting to these very heavy values. We can plot the same data in a cross plot, and I call this the diagenetic fingerprint. So here we have strontium on the y-axis and calcium on the x-axis. So our fluid buffered carbonates have a high calcium isotope value and a low strontium concentration, whereas our sediment buffered carbonates have a lower calcium isotope value. So they're essentially retaining some of that original value from aragonite, although they're not aragonite anymore. And then the primary aragonite is up here. So this is the sediments that have not yet recrystallized. So how can we use this fingerprint to think about reconstructing seawater, so the calcium isotope value of seawater through time? So I want to take you through an example here. So I'm showing you some data from Archean carbonates. So these are around 2.8 billion years old. They're definitely recrystallized. There's no primary aragonite left here. And the first thing is that we do see this fingerprint where we have this relationship between calcium isotopes and strontium concentrations. And I'm here showing you strontium to calcium ratios on a log scale because these Archean carbonates have pretty low strontium. So we can identify our fluid and sediment buffered end members here from this plot. We have our fluid buffered end member here to the right and our sediment buffered end members here to the left. And the Bahamas data I'm also showing here in gray for comparison. And one thing you'll see is that our Archean data is showing the same kind of trend, but it's consistently offset towards lower calcium and lower strontium concentrations. If we look at the modern data, we know that modern seawater has a value of 0 per mil. Modern aragonite has a value of minus 1.5, which fits this aragonite fractionation factor. We can do the same thing for our Archean data. So keeping this aragonite fractionation factor 1.5, we can move this over to our most sediment buffered end member. Adding the 1.5 per mil will then give us the seawater value of Meso-Archean seawater. So it seems like Archean seawater is around half per mil lower in the calcium isotope values. So we can use this to reconstruct calcium isotopes in seawater through time, question mark. So here I have made a timeline, and we're basically going to plot on some of these seawater values by using this fingerprint to reconstruct the seawater value. So first I've just plotted on the coral record from the Cenozoic I showed you before. Then I've gone through the literature and looked at some of the different studies that look at calcium isotopes. So this is a study from the Triassic-Jurassic boundary, where we also see this correlation between strontium and calcium ratios. And so I plotted the range we see in calcium here at the Triassic-Jurassic boundary. In this specific example, we don't see this full 1.5 per mil range in the data. So I have put a question mark here because I'm not quite sure if we're actually capturing the most sediment buffered end member in this case, although it seems like we do have some high strontium concentrations. Plotting on another study here from the Permian-Triassic boundary, where we have a data set that does capture this full 1.5 per mil. So we can plot on our data, and the blue dot here would indicate what I would say is seawater at this time, so minus 0.5 per mil. We can do the same for the Ordovician and the Silurian. Here, again, we don't see the full range, so I haven't plotted on a blue dot in this case. The Cambrian boundary, we do have some data that suggests that seawater is minus 0.5 per mil. The Ediacaran, this is the Schirrmonica. We also get to minus 0.5 per mil. Marinoan cap carbonates, the Trasona anomaly, so before the Marinoan glaciation. Again, we are sitting at seawater around minus 0.5 per mil. This is a study that compiled a bunch of different carbonates from the Precambrian that I plotted on as well. None of these really show the full range, but it still gives us some information about sort of where we are in this fingerprint space. And then finally, I have plotted on the Meso-Archean carbonates I showed you before, but also it seems like seawater is minus 0.5 per mil. So if we are to trust this record that's generated from looking at this fingerprint, it seems like the calcium isotope of seawater broadly has remained at minus 0.1 per mil relative to the modern from the Meso-Archean and all the way to the Cenozoic. And we really only see this increase in the last 65 million years. So what are the implications here? So we have to rethink, go back to the beginning of the lecture and rethink about our calcium isotope mass balance. So I put the equation that we derived for our steady state here. And we can take this now a step further and think about this fractionation of our our calcium isotope values in the bulk carbonate sink perhaps should be split into two different sinks. We have our fluid buffered sink, and we also have our rock buffered sinks. Some of these carbonates will retain their aragonite value, for example, whereas others would be diagenetically altered and have much higher calcium isotope values. So we can make a new model, assuming steady state, and we can split the mass balance equation into two sinks. So I've done that here. So we have our inputs equals to the fraction of carbonate sediments that are buried as sediment buffered plus the fraction that's buried as fluid buffered. And then we can rearrange this equation and solve for seawater. If we do that, we can make a new figure that shows on the y-axis the calcium isotope value of seawater, and then on the x-axis we have the fraction of carbonates that are buried as sediment buffered versus fluid buffered. Modern seawater, here's 0 per mil, would indicate that in the modern, around 65% of all carbonates are buried as sediment buffered. And this would be consistent with having a pelagic sinks where a lot of our carbonates are deposited on the deep seafloor, and they're probably not subject to as much fluid flow and diagenesis as carbonates that are buried in shallow carbonate platforms, for example, in the Archean, where this would indicate that only 30% are buried as sediment buffered. So I want to finish off this lecture by just looking at this compilation here from Peters and Hassan that is showing the proportion of carbonate sediments through time, where in the Mesozoic, we have the evolution of the pelagic sink in the deep sea. And if we go back in time, our bulk carbonate sink is mostly shallow marine carbonates. So I would argue that calcium isotopes can be a really useful tool for investigating the evolution of the carbonate factory through time. And we can actually potentially reconstruct seawater values, although we have to think a lot about diagenesis along the way. And that's the end. So I'm leaving you off with this photo that was taken a few months ago when we installed our multi-collector ICP-MS here at UVic. And just the day it was installed, we had this magical rainbow just above our observatory here, which has a happy smiley face as well. And I'm happy to take any questions.
Great. Thank you so much. That was really awesome. I learned so much, and the explanations on mass balance and so forth were so clear. That was really wonderful. I appreciate it. People want to just shout out with questions, or you can type it into the chat or raise your hand. Maybe I'll jump off with a little question.